All right, let's take a deep dive into this research article about bovine spastic syndrome.
SS.
Yeah, SS in Holstein cattle.
Yes.
It's pretty in-depth.
It is. It's fascinating.
This paper really digs into the genetics behind SS.
It does.
Which, for our listeners, it's a progressive neuromuscular disorder that resembles certain forms of hereditary spastic paraplegia in humans.
HSP.
HSP,
Yeah.
right? And they used whole genome sequencing, WGS,
Right.
to try and unravel the genetic mystery
They did. Yeah.
behind SS in these 7 affected Holstein cattle.
Yeah. Very cool stuff.
So I guess the best place to start is, what did they actually observe
Yeah.
in these cattle?
Yeah.
Clinically, what do they see?
So the article detailed...
And there's a range, right?
There is. Yeah, absolutely.
That's fun.
Table 1 in the article outlines the clinical signs observed in each of the 7 cases. And yeah, it really varies.
So they all had a progressive spastic gait.
They did. They all did.
But then beyond that,
Then it gets...
kind of a mixed bag.
Yeah. Some experienced intermittent spasms of the skeletal muscles of the pelvic girdle, while others showed kyphosis, a hunched back with their hind limbs extended caudally, and some also showed spastic flexion of the hind limbs with tremors.
Muscle biopsies.
Yes.
They took those too, right?
Figure 2 shows moderate paramycial and endomycial fibrosis
Okay, so fibrosis.
in these biopsies. Essentially, there's scarring in the connective tissue surrounding muscle fibers, which suggests muscle damage and degeneration,
That makes sense.
which aligns with the spasms and stiffness.
So that all kind of comes together.
Yeah.
But the big question is the genetics, right?
Yeah, right.
What did they actually find?
Well, this is where things get interesting.
Go.
They initially hypothesized that a single recessive gene was responsible for SS. However, their initial analysis, assuming a single recessive genetic cause, didn't yield any conclusive results.
So what did they do? They didn't just give up, did they?
No, they shifted their focus
Okay.
to explore other possibilities. Interestingly, in one case, case 7,
Yep.
they did identify a homozygous missense variant in the TOR3A gene.
Okay, so break that down for me.
Sure.
Homozygous missense variant, TOR3A.
This variant is particularly interesting because it results in an amino acid exchange in a highly conserved region of the TOR3A protein.
Gotcha.
Figure 3 illustrates the location of this variant and the affected amino acids.
And a highly conserved region, that means it's important, right?
Yeah, because it implies that this region plays a crucial role in the protein's function. Disruptions in highly conserved regions often lead to significant functional consequences.
Makes sense.
And the researchers suggest this particular variant could be deleterious,
Okay.
meaning it could potentially disrupt the normal function of the TO3A protein.
And this TOR3A gene
Yeah.
sounds familiar.
Yeah.
Didn't you mention something about a connection to human health?
You're right.
I feel like we've talked about this before.
Mutations in the human version of this gene, TOR1A, are associated with a specific form of dystonia.
Okay, and those both involve movement disorders.
Precisely. So this finding in the TOR3A gene could be a valuable clue in understanding the development of both conditions.
Yeah, very interesting.
Additionally,
Yeah.
the researchers looked at the frequency of this TO3A variant in various cattle populations. Table 3 shows that while it was present in various breeds, it was never found in the homozygous state except in case 7.
Oh, interesting.
This finding further supports the idea that this particular TR3A variant might be recessive.
So 2 copies are necessary.
Seems to be.
But I'm guessing
Yeah.
there's more to the story, right?
Exactly. The researchers didn't stop at the recessive variant.
Yeah.
They also investigated the possibility of dominant variants playing a role in SS.
So why did they even look at dominant inheritance at all?
Remember, they couldn't find a single recessive variant shared by all 7 cases.
Right.
This suggested that there might be other genetic mechanisms at play.
Makes sense.
So they shifted their focus to look for heterozygous variants,
Okay.
meaning variants present in only one copy of the gene,
Gotcha.
which could potentially exert a dominant effect.
Okay, so did they find any of those?
They did. In the remaining 6 cases of SS, they identified 7 heterozygous, likely deleterious variants in 6 different genes.
Okay, so what are the genes?
These genes are MPEG1, LHX8, OMM, NGRN, TTN, ATP1A1, and PCDH1.
Whoa. Okay, so what can you tell me about these genes? How do they relate to SS?
Many of these genes have known functions related to neuromuscular function or development.
Okay.
Figure 4 provides a visual overview of the locations of these genes within the cattle genome.
Okay.
For example, TTN codes for titin, which is a massive protein essential for the structural integrity of muscle fibers. And Table 5 provides a detailed breakdown of each gene, its function, and its potential link to neuromuscular disorders.
Gotcha.
And mutations in TTN are actually associated with various muscular dystrophies in humans.
So that seems like a pretty good candidate.
It is, yeah.
Great.
It hints at a possible link between bovine SS and human muscle disorders.
Very interesting.
Additionally, Case 3 had 2 potential dominant variants, one in WAM and one in NGRN, highlighting the possibility of digenic inheritance or individual effects these variants may have.
So this is getting pretty complex.
It is.
Right. We've got recessive variants, dominant variants, digenic inheritance.
Right.
This does not seem like a simple single gene disorder.
You're absolutely right.
At all.
This study suggests that SS in cattle has a complex and heterogeneous genetic basis.
And if I remember correctly, this also contradicts some previous research, right?
Yes, previous studies suggested other candidate genes for SS, such as FYN and CAXNA1A, which weren't found in these cases.
So our understanding of SS, it's still evolving.
It is. It highlights the evolving nature of scientific understanding and the need for continued research.
Okay, so we've gone through the symptoms, the genetics.
Yeah.
But what does this all mean?
That's a great question.
What are the implications of these findings?
And it's one we'll explore further in the next part of our deep dive.
Right.
We'll delve deeper into the specific genes involved and their potential roles in this complex neuromuscular puzzle.
Sounds good.
We'll also discuss the broader implications of this research, including its potential impact on our understanding of similar human conditions like HSP.
Awesome. I'm excited to learn more about that.
Me too. Let's dive a little deeper into some of these genes, starting with TOR3A. As we discussed earlier, this gene codes for torsin 3A, a protein involved in ATP binding.
Right.
Which, you know, that's crucial for cellular energy.
It's how the cells get their energy.
Exactly. And it's interesting to note that torsin 3A is found in high concentrations in neuromuscular tissue, suggesting a critical role in muscle and nerve function.
So it's definitely important in this area.
Yeah, for sure. And as you remember, the variant identified in Case 7 is located in a highly conserved region of this protein.
Right, and that's important.
It is.
Right.
Yeah, highly conserved regions of a protein tend to remain relatively unchanged across species throughout evolution.
So it's got to be important if it's staying the same.
Right. This suggests that these regions are vital for the protein's structure or function. So any disruption to a highly conserved region can have significant consequences.
Makes sense.
In this case, the missense variant in TOR3A is predicted to be deleterious, meaning it could disrupt the protein's normal function.
So it all kind of ties back to that human connection too, right?
Yes.
With the dystonia.
You're thinking of TOR1A. Yes, and it's similar to bovine SS.
Because they both involve movement, right?
It's abnormal movement.
That's certainly a plausible hypothesis.
Yeah.
Further research is needed to confirm this link.
Sure.
But it's a compelling avenue for investigation.
Okay, so moving on from TOR3A.
Yeah.
Let's talk about some of these dominant variants.
Okay, which one? Well, the TTN gene. That one seems pretty interesting. Because it codes for titin, which is this giant protein, right?
A giant protein crucial for muscle fiber structure and elasticity.
Yeah, and I remember that mutations in TTN can actually cause muscular dystrophy in people, right?
You are correct. Mutations in TTN are known to cause a variety of muscle disorders in humans, including several types of muscular dystrophy.
And those involve progressive muscle weakness.
They do.
Wasting.
Yeah.
Which we also see in SS.
Exactly.
So there are some parallels there.
There are some interesting parallels.
Even though the symptoms aren't exactly the same.
Yeah, particularly the progressive muscle degeneration.
Right.
The presence of this TTN variant in one of the SS cases definitely warrants further investigation.
So that's another good one to look into.
Yeah.
What about ATP1A1?
Okay.
That one seemed interesting too because it's involved with the sodium-potassium pump.
It is.
Which is like super important.
Yes, the sodium-potassium pump is a crucial player in maintaining the balance of ions across cell membranes.
And that's important for like
it is
nerve impulses.
Yes, this balance is essential for transmitting signals along nerves and triggering muscle contraction.
Muscle contractions, right?
If ATP1A1 isn't functioning correctly, it can disrupt this delicate balance, leading to problems with neuromuscular signaling.
And that also has a connection to human health, right?
It does.
With HSP.
Yes, mutations in ATP1A1 have been linked to certain forms of hereditary spastic paraplegia.
Right. And that also causes weakness and spasticity.
Yeah, the parallels between the findings in cattle and human conditions like HSP are quite striking.
Yeah, seems like a lot of these have connections to HSP.
It suggests that there might be shared underlying mechanisms at play, making bovine SS a potentially valuable model for studying these human disorders.
Okay, and then there was one that was a little bit different.
Yeah.
MPEG-1.
Okay.
Because that's involved in the immune system, right?
That's right, it's involved in the immune system.
But this is a neuromuscular disorder.
It is.
Why would that gene be involved?
That's a great question.
Seems a little strange.
It highlights the potential complexity of SS,
Oh.
while the exact role of MPEG-1 in SS is unclear.
Okay.
It raises the possibility of immune-related mechanisms being involved.
Okay.
There are known human neuromuscular disorders that are immune-mediated.
So the immune system is actually...
Yes, meaning they are caused by the immune system attacking components of the nervous system or muscles.
attacking the body.
The MPEG-1 variant could potentially be triggering a similar immune response in cattle,
Interesting.
leading to muscle damage and contributing to the SS symptoms.
So it's a lot more complicated
It is.
than we initially thought.
Yeah, we might be looking at a more multifaceted picture than just simple muscle degeneration.
Okay, what about these last few genes? LHX8, PCDH1, and NGRN. What do those do?
LHX8 is a transcription factor involved in the development and differentiation of neurons. If LHX8 isn't functioning correctly, it could disrupt the communication between nerves and muscles, leading to problems with muscle control.
Oh.
PCDH1, on the other hand, is involved in cell-to-cell adhesion, particularly in the nervous system.
Okay.
Disruptions in PCDH1 could potentially interfere with neuronal connections and signaling.
And NGRN.
NGRN is highly expressed in neuromuscular tissue and plays a role in mitochondrial ribosome biogenesis.
Okay.
Essentially, it helps ensure that the mitochondria, the powerhouses of the cells, are functioning properly.
Right.
The variant identified in NGRN could potentially disrupt mitochondrial function, which could have downstream effects on muscle health.
So it seems like each of these genes could potentially contribute to the development of SS,
Precisely.
even though they're all very different.
They are very different. This study really highlights the complex genetic landscape of SS and underscores the fact that it's not a simple single gene disorder.
Yeah, a lot more complicated than we initially thought.
It's likely a result of a complex interplay of multiple genetic factors, both recessive and dominant.
Which, again,
Yeah.
kind of goes against some of that previous research
Right.
that had pointed to different genes.
It does. This underscores the constantly evolving nature of scientific understanding.
Yeah, science is always changing.
It is.
Yeah.
New research often challenges existing paradigms and leads to a more refined understanding of complex diseases like SS.
So this is all great
It is.
and very interesting.
Yeah.
But where do we go from here?
That's the question, isn't it?
What are the next steps?
That's what we'll discuss in more detail in the next part of our deep dive.
Okay.
We'll explore the implications of these findings, both for veterinary medicine and for our understanding of similar human conditions like HSP.
Okay.
We'll also delve into the limitations of the study and highlight some avenues for future research.
Awesome. I'm really interested to hear more about that.
Me too.
We've spent a lot of time talking about the genetics of SS.
We have.
But what I'm curious about now
Yeah.
is what does it all mean?
Yeah, that's a good question.
Like, what are the implications
Well,
of all of this research?
this research has the potential to impact our understanding of human conditions as well.
Okay. Human conditions.
Specifically, those like hereditary spastic paraplegias.
HSPs.
HSPs, yes. As we talked about earlier, there are some striking similarities between the symptoms of SS in cattle and those of HSP in humans.
So we can learn about HSP
Yeah.
by studying SS in cattle,
Yeah.
potentially.
It's a great point.
Right.
Essentially, we can use cattle as a model system
Okay.
to gain a better understanding of human disease.
And cattle are good for that
They are.
because they're bigger than mice.
Yeah, cattle offer some unique advantages for
Yeah.
this type of research.
So it's easier to study them.
Well, for starters, cattle are larger than typical lab animals like mice, which can make it easier to study certain physiological processes.
Right. And they live longer too, right?
Yeah. Additionally, their longer lifespan allows researchers to observe the progression of diseases over a longer period.
And we already breed them.
Exactly. And since we already breed and manage cattle for agricultural purposes, there's access to large populations and detailed records, which is invaluable for genetic studies.
So it has implications for both
It does.
veterinary and human medicine.
For both, yeah.
So starting with the veterinary side,
Okay.
how can this help with cattle health?
Understanding the genetics of SS could lead to better diagnostic tools for this condition.
Okay.
Imagine being able to test cattle for these specific genetic variants to identify those at risk of developing SS.
So we could test them
Yeah,
before they even get sick.
that would be a game changer
Right.
for breeders,
So it would be useful for breeding programs.
allowing them to make informed decisions about breeding practices, potentially reducing the incidence of SS in future generations.
And what about the human side?
Yeah,
Could this lead to new treatments
potentially.
for HSP?
That's the real question. If we can pinpoint shared genetic pathways or mechanisms between SS and HSPs, it could pave the way for the development of new therapeutic targets for these human conditions.
So this could eventually lead to treatments for people.
Yeah.
But humans are more complicated,
Like, absolutely right.
than cattle.
Human genetics is obviously far more complex than cattle genetics.
So it's not a guarantee
No.
that this will definitely lead to treatment.
There are likely many other factors, both genetic and environmental, that contribute to the development of HSPs.
But it's a good start.
So while the study is promising, there's still a long road ahead before these findings translate into treatments for humans.
And another thing that I thought was interesting was how complex it all was. Genetically.
Yeah.
It wasn't just one gene.
It's a great observation, and it highlights the intricate interplay of genes and complex disorders.
Right. It was recessive, dominant, digenic.
We're moving away from the simplistic one gene, one disease paradigm as we delve deeper into the complexities of the genome.
So it's not as simple as we thought.
It seems that many diseases, like SS, arise from the interplay of multiple genetic and environmental factors.
So we need to look at the big picture.
This study serves as a reminder that embracing this complexity is crucial if we truly want to understand and treat these challenging conditions.
Well, this has been fascinating.
It has, yeah.
I've learned a lot.
This research has opened up a whole new chapter in our understanding of SS and its implications for both veterinary and human medicine.
It seems like this research is just the beginning
It is.
of a lot more to come.
It underscores the power of scientific inquiry and the importance of cross-disciplinary collaboration in pushing the boundaries of knowledge. From Vet Neurojar, keep those minds inspired, hearts light, and tails wagging.